Hello, everyone. Thank you for joining. We're going to give it just a couple of minutes and get started right about 12:02. All right, I think we have just a few trickling through the waiting room, but we'll go ahead and get started today. So, hello, welcome. Thank you guys for joining. My name is Carson Patton, and I am the Events and Programming Manager at the UNC Nutrition Research Institute here in Kannapolis, North Carolina. We are glad that you could join us today for our Appetite for Life event exploring how folate works. Appetite for Life is our monthly series that we designed to bring nutrition research directly to you. So each event features experts from the NRI who translate their scientific work into everyday insights that you can use to better understand your health. Vitamins are widely perceived as beneficial across the board, which has fueled this idea that more is automatically better. But nutrition science tells us a more nuanced story. How much we consume, the form our nutrients take, and how our bodies process them all matter, sometimes in ways that are not so obvious from supplements, labels, or their headlines. So today's discussion invites us to look more closely at folate, which is a B vitamin essential for health across the lifespan and especially well known for its role in early development. At the same time, ongoing research raises important questions about excess intake and the differences between naturally occurring folates in food and synthetic folic acid, which is found in supplements and fortified products. So our first speaker today is Natalia Krupenko. Dr. Krupenko is an associate professor in the Department of Nutrition at UNC Chapel Hill. She's been part of the UNC Nutrition Research Institute faculty since 2014, and her research focuses on how folate supports human health and how imbalances may contribute to disease risk. She'll be joined by Madeline Childress, a graduate student in the Krupenko lab. Madeline joined the NRI in 2019 and works with Dr. Natalia Krupenko to research the role of folate in promoting human health and preventing disease. We encourage you to submit your questions throughout the presentation using the Q&A feature or the chat feature, and we're going to address as many as we can during this discussion portion at the end. So with that, I'm pleased to turn things over to Dr. Krupenko.
Thank you, Carson. So my slide is visible well, yes?
Yes, you're good.
Perfect. Thank you. Well, that's my favorite talk about folate, and I will start just with a reminder of what vitamin is. Vitamin is the organic compound that is required by the body, sometimes in tiniest amounts, but it is absolutely necessary. Even if it is deficient, it will result, the body will get serious disease. So it is vital nutrients. Folate has been discovered relatively later than other vitamins by these British scientists, Lucy Wills, who went to India to study how to deal with the macrocytic anemia that was very highly prevalent in young women at the textile factories. She worked really hard on it, and she found out that apparently yeast extract could correct the anemia. Then she discovered that liver extracts can also help with that, and it took years. It wasn't the fast process. Finally, when in 1941, chemists isolated a compound from spinach and called it folic acid, she asked them to share their compound and test on the women whether it also has the protective effect. In fact, it did, and that's how the Wills factor became the folic acid, the necessary vitamin. The structure was very soon confirmed by the chemical synthesis, and only 5 years after the vitamin was exactly established as a vitamin, American physician Sidney Farber used synthetic antagonists of this folic acid to treat leukemia, which shows how fast the importance of the vitamin was used for battling the cancer cells. So these formulas shown here are showing folic acid up here. I would like to reiterate that folic acid, despite widely called vitamin, it is not strictly a vitamin. It cannot perform vitamin's function of binding and donating one carbon groups. It is provitamin. It's a precursor of the vitamin, which after reduction is a process of adding hydrogen atoms, and 4 hydrogen atoms are added right here. Only in this form, the molecule can accept or donate one carbon groups. So the vitamin is tetrahydrofolic acid, provitamin is folic acid. And of course, all of the derivatives. And on the right, I show that it's not just one carbon group of a single structure. It's 5 different carbon groups, which makes us understand that there are different forms of the vitamin with different one carbon groups. So these one carbon groups are used to add, for example, methyl group to a small molecule, making new molecules. Adding the same methyl group to DNA, RNA, or protein is a regulatory effect. And the formyl groups, which are used for RNA and DNA nucleotide biosynthesis, are supporting growth and division of cells. And that is the reason why the vitamin has been implicated in prevention of neural tube defects. It was actually British Medical Council which performed a trial with the pregnant women who had previously neural tube defects affected pregnancies. And in this trial, they have shown that those who received vitamin B9 or folic acid, it's another name for folic acid, folic acid in combination with other B vitamins, those women had significantly 70% lower frequency of neural tube defects, while the group which did not receive the vitamins was affected much stronger. So the recommendations were created for all women of reproductive age to consume between 400 micrograms or 0.4 milligram to 1 milligram of the vitamin daily. And those who had previously neural tube defect affected pregnancy should take even 4 milligrams per day. Well, this is a good recommendation, but there are issues. 50% of pregnancies are unplanned, and neural tube closes very, very early in the prenatal development when women often don't even know that they are pregnant, so they cannot start taking vitamins. 2/3 of women reported inadequate folic acid. 1/3 of women reported taking a multivitamin with folic acid. 2/3 did not. And 1/3 of all women and half of all unplanned pregnancies do not even start prenatal care in the first trimester, which means these women, even if they start taking vitamins, would be too late. So the FDA in the United States has developed collaboration with the scientists, and on March 5, 1996, they issued an statement that the FDA is introducing mandatory fortification of flour, breads, and other grains with a folic acid so that everybody will be taking at least some folic acid. I want to note here that the scientists calculated the fortification amount in such a way that average American diet would add 129 micrograms of folic acid to whatever the person is already taking from the natural foods. And this amount is not overly high. It was developed in such a way that it won't overload the consumption, the amount of folic acid taken by the person. However, not only pregnant women are taking these products, but entire population, and the whole population is affected. Well, due to the success of the British medical trial, and the success of that was observed after food fortification in the US, which reduced NTD-affected births by 20-40%. A lot of other countries started doing that. And in 2024, we count 82 countries that are supplementing. They're all shown here in the dark blue. But that means that there are a lot of people who are exposed to the folic acid, and not necessarily all of them are women. At least half of them are supposed to be men. So another aspect of this which causes concern is the fact that people are using multivitamin supplements. And they are using multivitamin supplements not because they are all deficient, but rather to improve overall health. And that could create a situation when the intakes of some of the vitamins and folic acid specifically could be too high. The epidemiologists have been doing evaluation, and several studies found a relationship between fortification for using folate supplementation and colon cancer, breast cancer, prostate cancer, and lung cancer. It is not proven, but there is some correlation. So that creates a very interesting question. Should everybody take folate? And how safe are we in this respect? And our lab has run a study of high dietary folate effect on cancer metastasis. And I'm showing you the data here. I'm not going to go too much into detail. I will only tell that we used cancer cells that are modified to give luminescence under the UV light so that we can evaluate the amount of cancer cells in the live mice. And the mice were divided into two groups. One was kept on the restricted folate, which means no folic acid was added to the normal products that mice eat in vivarium, some grains, some starches, and so on. While the other high folate diet had six-fold higher level of folate than normally done in the normal diet. Now, we watched how these cells grew, and these circles up above the bar are showing the rate of growth, the volume of tumors on the restricted diet, and on the bottom, on the high folate diet. Nobody would like to see this in a patient. That is a very alarming result. Now, we also tracked the survival of these mice. And on the top, you see on restricted diet, the mice, 50% of mice survived by 9 weeks. Not so bad. Those that were on the unlimited folate diet, on the high folate diet, by 5 weeks, there were only 40% left. And even these died between 5 and 7 weeks. So that extremely important factor also affected the survival. In general, all of these falls into the category of more of a good thing is not always better. And everything, every nutrient, every biological factor is sort of found to have a U-shaped curve. If there is very little nutrients, there will be high risk of defects and death. Once the nutrients intakes are increased, the risk is going down, and finally it reaches the optimum when the body gets everything that it needs. So the risk is minimal. It stays minimal within certain interval, but then once you start consuming too much, the toxic levels start manifesting, and the risk of disease again increases. So doesn't matter what it is. It may be vitamin, it may be some sugar, it may be even water. Too much water also has the risk. So each nutrient does this. And that's another argument why we should be careful with supplementing. Well, after observing the manifestations with connection to cancer, we also wanted to see what metabolically happens with these mice. And this project was done by my former graduate student, Carrie Barron. It included 3 groups of animals which were placed on different diets. Again, one diet was restricted diet, which means that only what is present already in the food, no additional folic acid. Control diet had a little bit. This is the normal addition to any standard vivarium diet, which is 2 milligrams per kilogram of food, and then the 12 milligrams per kilogram of food was the high folate diet. And we isolated, we kept the animals on these diets for 4 weeks only, and then we tested the content of folate and different metabolites in the livers of these mice. We actually done metabolomics, which means measurement of all metabolites in the liver. It gives a comprehensive snapshot of what is happening there. So, as we expected, the more folic acid in the diet, the higher levels of folic acid are in the liver. The same was true for the first step, where folic acid is converted into on the way to the biologically active compound. And here we also have seen increase with the increased absorption supply. However, for the storage form of folate, 5-methyl-THF in the liver, we were surprised to see that it is quite the opposite. The more folic acid you give, the less is the storage form. How can it be? Well, these mechanisms are not quite well investigated, but we already have some indications of what effects the high folic acid has that reduces the vitamins. Again, in the peripheral tissues, the increase was the proper. However, it is happening because liver handles all folate that we ingest. it converts it to 5-methyl THF and only then releases into the tissues. And what surprised us even more was the fact that B vitamins, B1, B2, B6, which were supplied in exactly the same amount to all groups, were also behaving like the storage form of folate. You can see this downward trends in all groups for the active forms of the vitamins. We have not observed this before, and nobody has published that yet. But we are really looking forward to figure out what's happening and why this is happening. There is another quality of supplementation with folic acid, and it is possibility to mask vitamin B12 deficiency. If the person is vitamin B12 deficient, and all of this is manifested as anemia, but folate deficiency also shows anemia. And if the person doesn't test for the patient for the B12, then giving just the folic acid will correct pernicious anemia. It will disappear, but the fact that B12 is deficient will not go away. And B12 is critical for brain function. And that will lead to the fact that the patient will remain deficient and his health will deteriorate. So the summary of what we have found with these experiments are listed here. It shows that folic acid depletes liver folate, that excess affects other vitamins, B vitamins, and even the fat-soluble vitamin. And the next question we are working on is, are these effects characteristic for folic acid only, or other natural vitamins do that also? We don't know that, but we believe that the main approach that can be used in this situation is to use balance. Do not overuse, do not exceed anything. And this was kind of stated by the USDA and Department of Health, Human Health Services in the dietary guidelines for Americans, that you should try to get vitamins primarily from food. And the food usually contains additional factors that help you to maintain health. Additionally, if you want to make sure that you are getting enough vitamins from food, try to preserve them. Try not to overcook vegetables. Try to eat vegetables raw, steam fry or stir fry the vegetables. And one of the interesting products that contain folate, but preserve it much better than the fruits and vegetables, is beans and peas. When they're cooked, even for a long time, they preserve their folate a lot better than the fresh fruit and vegetables. And this little figure shows you that you can get half your daily recommended daily value from spinach, asparagus, and from a cup of lentil, you get almost entire your daily value. So to supply the vitamins from the food in sufficient amount is not that difficult. It's easy. And this is actually the cover of the new dietary folate recommendations for Americans from 2025-2030. I'm really glad to see that this new dietary guidelines focus on the diversity of food, on the mixing all of foods, not on excluding something, but it states that everything should be used in amounts appropriate for individual. So they are focusing on individual nutrition requirements and on the diverse mix of the products that should be providing enough for the health. Now, regarding the properties of natural folate and synthetic folic acid, I would like to show you these bottles. They demonstrate that currently pharmacies have 3 different forms of folate supplements available. Methylfolate, this is the natural one. 5-formyl folate, also close to the natural one, and folic acid. And if indeed different forms have different effect. A single dose of 5-methyl-THF increases blood folate concentration much more efficiently than folic acid. And a special attention right now is seen in the press and in science is regarding the 5-formyl tetrahydrofolate. This form has been used to bring to treat the cerebral folate deficiency. Cerebral folate deficiency has been linked to the autism and some other neural disorders. And it was shown that when people were given 5-formyl tetrahydrofolate, the cerebrospinal fluid folate normalized, if not completely, but was brought closer to normal range. We do not know yet whether 5-methyl-THF can do that, and only for one reason. There are no parenteral preparations right now to test this. But we are expecting that it should be the same. However, the folic acid, after certain experiments and certain attempts to treat the patients with the cerebral folate deficiency, it is actually not contraindicated, but it's not recommended. The reason for that is that folic acid binds to one of the carriers of folate. The brain has 3 different folate carriers: reduced folate carrier, proton-coupled folate transporter, and folate receptor alpha. And the folate receptor binds very tightly. It gets into the cerebrospinal fluid, but it does not release folic acid, and it cannot go into the cells. Some drugs also can affect this. So yes, indeed, it matters what form you are taking. And in the end, I would like to say that we are studying, we're learning a lot more every day. But in general, if you look at the approach for nutrition, the old statements provided by the father of the American Nutrition Research and Education, Wilbur Olin Atwater, they cover all aspects of what we're learning right now. When asked what are the main principles of the healthy nutrition, this person who studied nutrients, who studied nutrition, answered there are 3: variety, balance, and moderation. And that applies to every single aspect of what I've told you today. So on this, I'm done. Thank you. Sorry, I took a little longer.
Do you want to kill your screen sharing?
Yes, I completely forgot. Can you share now?
Yes. Let's see. Is that good, or are you seeing?
You're good, Madeline.
Okay. Excellent. So yes, I'm Madeline Childress. I'm a PhD candidate under Dr. Natalia Krupenko. And I'm going to do a little bit of a shift here. So Natalia just talked to us about what folate is, and I'll shift to what does studying folate look like, and talk a little bit about my PhD project. But before I dive in, I want to briefly reiterate some of what we just heard because it's really the foundation for everything that I study. So you can think about folate like fuel for your cells, not fuel like calories or sugar, but fuel like the parts and tools your engine needs to run. Every cell in your body is constantly growing, repairing itself, and making new DNA, and folate is absolutely essential for that. So if you don't have enough folate, your cells kind of act like a broken down car. Necessary molecules aren't made, things start to malfunction. Over time, that actually increases disease risk, including cancer. But if you have the right amount, now you're kind of like a reliable family car. Everything runs smoothly, cells grow and repair exactly like they should. And then over here is where things get interesting. Cancer doesn't play by the same rules as our normal cells. Cancer cells are more like race cars. They're growing fast, dividing nonstop, and burning through resources. So while folate is healthy and necessary for normal cells, if cancer is already present, that same folate can actually help tumors grow faster. It's like giving the race car premium gas. So in cancer biology, folate is known as a double-edged sword. It's essential for life, but potentially dangerous in the wrong context. So if folate is the fuel, what is it actually powering? And again, some of this is review, but cancer by definition is when some of the body's cells grow uncontrollably. They divide over and over, and every time a cell divides, it has to copy its entire DNA. That's all 3 billion letters of it, and you can think of that almost like photocopying an entire encyclopedia every couple of hours. So to do this, cancer cells become really hungry, not for calories, but for the building materials. Folate is one of these materials. It's essential for creating DNA, and it also powers something called one-carbon metabolism. Think of one-carbon metabolism kind of like a factory assembly line. It moves tiny chemical pieces, or single carbon atoms, around the cell. These pieces are used to build DNA, amino acids, lipids, and other cellular components needed for survival. One-carbon groups can also be used to regulate DNA and proteins, which helps cancer overtake a cell. Because cancer cells divide so fast, they depend on this system even more than normal cells. They're extra hungry, which fortunately means they're also extra vulnerable. If that system shuts down, the cell can't build what it needs, and if it can't build DNA, it can't divide. This gives us a tool to use against cancer. So we commonly use a class of drugs called anti-metabolites. These drugs interfere with the nutrients cancer cells rely on to grow. And one of the most common types, and actually the first ever chemotherapeutic developed, are called antifolates, drugs like methotrexate or pemetrexed. And these work by kind of essentially poisoning the fuel line. The drugs mimic the structure of folate, but they don't work. Our cells can't use them. They're a decoy. It's like putting a fake key in the lock. Now, folate can't fit because the enzymes that use folate are jammed up. Cell can't use folate, and as we've seen, without folate, the cell can't grow and divide. So then it goes into crisis. Stress signals turn on, which is basically an internal alarm system. And at this point, the cell has a choice to make. It can either repair itself or self-destruct. In cancer therapy, that shutdown is exactly what we're hoping for. So what happens in the cell after we cut off folate? Cell doesn't just immediately die. First, it kind of panics, that alarm system I mentioned. This is something we call cellular stress, and stress doesn't necessarily mean damage yet. It's basically the cell's version of a smoke detector. It's a warning system that says, something's wrong, we need to fix this now. And cells use stress responses all the time to protect themselves. For example, if DNA gets damaged, stress turns on the repair systems. If nutrients or oxygen run low, stress cells growth to conserve energy. And if toxins appear, stress activates those cleanup pathways. So in normal life, cellular stress can actually be helpful. It keeps cells alive, it buys time, it helps them adapt and survive. Constant or unresolved stress is, of course, harmful and can lead to aging and disease. But in the short term, cellular stress is an alarm system to trigger defense mechanisms. So in cancer therapy, we can intentionally trigger this stress. Our antifolate drugs create kind of a starvation effect. Suddenly the cell can't make DNA, it can't build what it needs, so all these alarm bells go off. And again, it has this decision. Do I fix the problem or do I shut myself down? And that decision is controlled by specific proteins inside the cell. And one of the most important of these proteins is called p53. p53 is one of the most powerful tumor suppressors in the human body. It's so important that scientists call it the guardian of the genome. You can think of p53 as the cell's emergency brake. If something goes wrong, DNA damage, low nutrients, toxic stress, those cellular stressors we just talked about, the alarm system turns p53 on. And p53 can do three really important things depending on the cause and the severity of the stress. First, it can pause growth, basically tells the cell, stop, don't divide right now. That gives the cell time to fix the problem. Second, it can activate repair systems. It turns on genes that patch damaged DNA and restore normal function. But if the damage is too severe, p53 kind of pulls the final lever. It tells the cell to self-destruct. This is a process called apoptosis. Now, it sounds a little dramatic, but it's actually protective. It prevents dangerous, abnormal cells from multiplying, or cancer. So when p53 is working, it's constantly preventing cancer before it even starts. But here's the problem. In over 50% of cancers, p53 is mutated. It's broken. That emergency brake is gone, so now damaged cells don't stop. They don't repair, they don't die, they just keep dividing. Errors pile up, and tumors grow. For decades, p53 has been considered what we call undruggable. This is because most cancer drugs work by blocking something that's overactive, just in the case with antifolates. But p53 isn't overactive, it's broken. In many cancers, the protein is unstable. It's there, it just can't hold its shape. So if it falls apart before it can do its job. And it's a lot harder to repair a broken brake than to just cut one. So once it's damaged or mutated, we haven't had a reliable way to restore it. This is where another key player comes in, a molecule called ceramide. Now, the term ceramide may sound familiar if you use CeraVe or any number of skincare brands. You've probably seen it on the ingredient list if you're a moisturizer. That's because ceramides are a type of lipid, basically a fat molecule. And most lipids act like building materials. They form the membranes that surround every cell. They help create barriers, keep things sealed, and give cells structure. In your skin, for example, ceramides help lock in moisture and protect the skin barrier, which is why those skincare companies love them. So for a long time, scientists thought ceramides were just structural, just kind of part of the wall. But over the last couple decades, we've learned that they're doing much, much more than that. Certain ceramides actually act as signaling molecules. They help cells respond to stress. They're part of that alarm system. And one specific type, called C16 ceramide, has been strongly linked to diseases like diabetes, neurodegeneration, and cancer. When C16 ceramide levels rise, it actually pushes the cells towards apoptosis, that programs cell death. So instead of just being a part of the membrane, it can act like a stress message that says, something is wrong, it might be time to shut down. And here in the cell, on the endoplasmic reticulum, or the ER, is where the cell actually makes that signal. There's an enzyme or protein called CERS6, ceramide synthase 6, whose job is basically to manufacture C16 ceramide. You can think of it as a little factory. When the cell is calm, it makes small amounts of ceramide. But when the cell is stressed, that factory ramps up production. It starts making a lot more ceramide. And here's the key connection to everything we've been talking about. These antifolate drugs, like methotrexate, the ones that starve cancer cells, create exactly the kind of stress that ramps up production. So when we treat cells with antifolates, CERS6 turns on, ceramide levels go up, and those death signals get stronger. So now we can finally put all the pieces together. So we started with antifolate drugs. Those drugs starve the cell of folate. That creates cellular stress. And we learned that stress activates two important players, ceramide, the stress signal, and p53, the decision maker. My research is really focused on how they actually talk to each other. First, stress increases ceramide production through CERS6, so ceramide levels rise. And ceramide doesn't just float around, it can actually bind directly to p53. That binding helps stabilize p53, almost like flipping it fully on. So now p53 is active. Once p53 is active, it goes to the cell's control center, or the nucleus, here at the bottom, and starts giving instructions. It turns on genes that help the cell respond to stress, and one of those instructions it gives is to make more CERS6, which means p53 tells the cell to make even more ceramide. So now we have a loop. Stress makes ceramide, ceramide stabilizes and enhances p53, p53 makes more ser6, which makes more ceramide. It's one big cycle. And that amplification pushes the cell past the point of repair and towards apoptosis, or death. So instead of a weak stress signal, the system basically says, this cell is too damaged, shut it down. So this stress ceramide p53 loop is what my research focuses on understanding and potentially exploiting to kill cancer cells more effectively. But to study that interaction, we needed a way to actually see these proteins meet inside the cells. How do you actually see that? Well, proteins are very, very tiny. You can't just watch them kind of bump into each other under a microscope. So we use a technique called BIFC, which stands for bimolecular fluorescence complementation. It sounds complicated, but the idea is actually really simple. We basically turn protein interactions into light. So we take a fluorescent protein, kind of like a very tiny light bulb, and we split it in half. By itself, each half is dark, no glow. Then we attach one half to p53 and the other half to ser6. Now, if these two proteins never meet in the cell, the halves stay separate, nothing lights up. But if p53 and ser6 physically interact, they bring the two halves together, the light bulb snaps back together, and the cell literally glows green. So no interaction, no light. Interaction, fluorescence. It's kind of like a molecular light switch. So this lets us directly see when and where these proteins interact in living cells. And that's one of the main tools we use to test whether stress, like anti-folate treatment, actually brings p53 and ser6 together. So here's what that looks like. Remember, in this assay, green means interaction. No green means they're separate. They're not talking. So let's start on the left. With no stress, the cells are basically dark. There's no green signal. You just see the nucleus, that command sender, in blue. So under normal conditions, these two proteins don't interact. But when we stress the cell, that changes. Whether we block folate with an anti-folate drug like methotrexate, or add a ceramide-based drug directly, or simply remove nutrients from the cell, we start seeing bright green fluorescence. Different ways to stress the cell through the folate pathway, same response. p53 and ser6 come together. p53 can acquire C16, that stabilizing lipid being made by ser6, and start apoptosis. And then we can make this a little more fun and add kind of any number of colors. Besides that green interaction signal here, I can label other structures too. So here I've dyed the endoplasmic reticulum in red. Remember, that's where ser6, our ceramide-producing factory, is located. So now we're not just asking if these proteins interact, but we can see where it happens. So green is the interaction, red is the ER, and when they overlap, you get yellow. So this interaction is happening exactly where the ceramide is being made. Now, we know that p53 and ser6 are meeting at the right place in the cell, but seeing it is one thing. What really matters is, can this pathway actually help us treat cancer? So up until now, everything I showed you in those BIFC pictures was cells with normal working p53, or what we call wild type. But remember the big problem we talked about earlier. Over half of cancers don't have normal p53, they have mutated p53. And mutant p53 has historically been considered undruggable. So the real question becomes, can we still kill those cancers? Does ceramide binding help target mutant p53? Here we tested 3 different types of cancer cells. So the green bars in both these graphs are cells with normal wild type p53. The purple and blue bars are cells with mutant p53. So this left graph is methotrexate, that anti-folate alone. You can see that the normal cells respond pretty well, but the mutant ones, they barely die. They're much more resistant. So anti-folates alone aren't enough to jumpstart this cancer-killing machine. But when we add a ceramide-based drug on the right with the methotrexate, something interesting happens. We see a much bigger drop in survival, even in the mutant p53 cells. So combining ceramide with methotrexate creates a stronger synergistic effect. It helps us target cells that normally wouldn't respond, which suggests that this ceramide pathway might be a way to enhance stress signaling even when p53 is mutated. And that's really the big picture of my work. So to wrap up, our BIFC imaging gives us a way to actually see when proteins interact inside living cells. Not just guess, not just infer, but literally watch it light up. And that lets us ask a few really specific questions. By using different variants of p53, we can see if mutations change this binding, which additional drugs might strengthen or block this interaction, where in the cell does this happen, and which parts of the two proteins matter the most. And answering those questions isn't just academic. It helps us understand how stress signals flow through the cell and how we might manipulate those signals to kill cancer cells more effectively. Or conversely, which cancers won't respond to this type of therapy. So by mapping the ceramide p53 folate pathway, we're hoping to find new ways to design smarter, more precise therapies, ones that turn the cell's own stress signals against the cancer. And that's the goal of my work. And I will end it there and kick it back to you, Carson.
Awesome. Thank you so much, Dr. Krupenko and Madeline. That was a super informative presentation. I especially love the fuel metaphor, Madeline. I thought that was really cool. So we're going to move on to our questions shortly, but I wanted to take a minute to wrap up with just a few notes. So get your questions ready. Go ahead and add those to our Q&A. And so today's presentation reinforces that nutrition is rarely about simple rules of more or less of a nutrient or a food group. Our NRI scientists encourage a balanced, contextual approach to diet, one that considers how individual nutrients function within the body. And NRI programs like Appetite for Life are designed to make current nutrition science accessible and relevant. If you enjoyed today's presentation and would like to stay connected, you can follow us on social media or sign up for our email list at uncnri.org to learn more about upcoming events and new research. After this webinar, when we close, you'll receive a brief survey, and we appreciate you taking a moment to share your feedback. It really helps us guide future programming. And if you're interested in supporting the NRI's work, like what's being done by Madeline and Dr. Krupenko, you can participate in research or help expand our reach, and you can find additional information at uncnri.org/support. And we will go ahead and move into our discussion portion. So thank you again for joining us today, and we hope to see you at future Appetite for Life events. I'll go ahead and get started with our questions. Okay, we were asked, as we know that folic acid is different than folate, some of us need methylfolate due to MTHFR changes. So what does folic acid do to us?
Well, it's a question for me. We did not test how additional effects of a folic acid would manifest in the case of the MTHFR deficient cells or people. These experiments are not so easy to do because it might present some risks. But with the MTHFR deficiency, again, there were cases that demonstrated that people, when they're given the folic acid, their function of MTHFR does not improve. They still have deficiencies in some folate forms and processes. However, when they were given 5-methyl-THF, that was able to fix the problem because you give the product which MTHFR is making. Because in the cells, the 5-methyl-THF is made by this enzyme MTHFR, which reduces the methynyl group that can be on the folate. So in this situation, people who have MTHFR deficiency, they definitely should take I'm not a physician, I cannot kind of recommend what not to take, but they should talk to their physician about the form of folate and the fact that 5-methyl-THF is a product of MTHFR, and it may be more beneficial to the person, more efficient working in the body. But make sure you talk to your doctor. This is the scientific aspect.
Yes, perfect. Another question, with so many enriched foods in the United States, how easy is it to exceed recommended folate intake without realizing that you're exceeding?
If you're not eating more than you're supposed to, if you're eating what your body is meant to eat, then you probably won't overwhelm your system, because, as I said, the calculation of how much to add to the grain foods was made based on what average American takes every day. Certain amount of bread, certain amount of grain, other grains, and so on. And plus, of course, the other food sources. But we are not average. All of us are slightly different. Somebody is having smaller weight, smaller body. They probably would get a little bit more of this over supplement than might be necessary for their body. So there is a big variability, and there are people who eat a lot. Then again, the more you eat, the more you get the additional supplementation with the folic acid. And it may not be dramatic if you are holding within that normal range, like the nutritional recommendations underscore. Every person has their own requirements. So you need to kind of see how you feel, how your body behaves when you are consuming these products. But just the supplementation in the food, the addition, it is definitely not good for people who might have cancer. But for the healthy person, they are not changing that much. However, if you're consuming more than necessary, then it's a different story.
Thank you. Let's see. As a general rule, should we avoid vitamins with percentages that are way over our daily recommended intake?
Well, ideally, you should consult your physician before you start taking multi-supplement vitamins, because if you are consuming enough, then taking more of it probably won't make you more healthy. But there's always a risk of having a negative effect. But yes, I would say that for most people, having double, triple daily requirements would not be very useful. However, as I mentioned, for the MTHFR impaired situations, in that case, maybe using a little more would be appropriate. So you need to consult your physician, or at least the certified nutritionist, because those people are trained in calculating your need, your what you're consuming, and that they will be able to evaluate will it overwhelm your system or not.
Perfect. So we have a question. If you have liver cancer and you're on chemo, what do you do about folic acid intake? As you mentioned, it may not be, those enriched folate foods may not be great for people who do have cancer. So I think that's where that question is coming from.
Yeah. One of the options would be to try find the non-fortified products. Honestly, I don't know how many of those can be found in the US, But for example, buying bread from the non-fortifying countries like German bread probably won't contain the folic acid. But then again, you probably need to talk to the nutritionist and kind of and the physician. I don't know how physicians are up to date with the whole information on the effects of folic acid, but in any case, even people who have cancer, they need folic acid because they, not folic acid, vitamin folate. So the natural folate would be the best option to support the normal function of the body.
I have my own personal question as we're within this this question thread. So, have we seen enough benefits to outweigh the risks of enriching food products? You mentioned that Germany doesn't enrich. I'm wondering if we've kind of decided that it's better than not.
Well, the data so far have not been collected in sufficient amount because yes, we see correlations, we see some effects, we see improvements. Yes, indeed, the percentage of babies born with a neural tube defects has dramatically reduced. The reduction was depending, of course, on the areas where the people live, but it was between 25 and up to 40%. And that is a highly important thing, and especially with the consideration that the baby born with this defect would be a tremendous burden on the parents, would be suffering on his own. It wouldn't be really good for the baby. And that also puts burden on the medical professionals to take care of these kids and try to bring them to more or less decent conditions. So in that case, yes. With regard to all of the others, we do need to consider that some people might not have advantage when they are eating these foods, and maybe there should be foods both with supplementation and not supplemented. And in that case, the person, based on their own health history, might be able to select what fits best for them with help. Again, I always say talk to your physician because that is a very, very strong indication that you need to have professional help in that.
Okay, we have time for just one more question. I just got a few. Let me read through them.
Maybe let's ask Madeline something.
Yes. Without speaking to a doctor or a nutritionist, are there things that we could look for as far as symptoms of high folate or low folate, and what might that look like?
You probably won't see any symptoms that you could register because it is really having low toxicity, but the biological regulatory effect, which manifests in speeding up division of cells, that is much more important. So I don't think that there are any symptoms that a person can register. It can be checked doing the measurement of folates in the at least in blood. Course, tissues is not a good option, but at least just following. But these tests are complex. It's not easy to measure folate, and especially folate forms. I know it firsthand because we do measure it in the lab. it's a very complex method. Clinics do not have it yet. But with the development of mass spectrometry, I believe they will start doing that soon, and hopefully it will be available for clinical applications. But for now, there's not much, and unless you eat, I don't know, 10 milligrams, no, 10 grams, which probably will cause diarrhea just because of the disbalance of the electrolytes in the digestive tract. So no, you cannot figure out whether you're eating more or less.
Perfect. All right, we're at time. So thank you all so much for attending. We hope that we'll see you at our future Appetite for Life events. Thank you.
Thank you.